Okay, I think we're going to get going. It's my pleasure to welcome Dr. Peter Gwynne as today's speaker. Pete did his undergraduate at the University of Cardiff and then did, and I'm quoting from his CV, a bespoke PhD at the University of Edinburgh, where he was farmed out to industry early on in his career and worked on developing antimicrobial devices. He joined us in Tufts 4 years ago now, 3-4 years ago. Yeah. And where he's been working on Lyme disease, and he has developed some really cool insights into lipid metabolism in Lyme. But I don't think that's what he's going to talk to you about today. He is actually going to talk to you, I think, about a project that developed as part of being locked down for the pandemic. So this is another bespoke project. Pete is now a research assistant professor here at Tufts. He is the current reigning champion of the Bay Area Lyme Disease Foundation Emerging Leader Award. Pete, welcome.
Thank you. Okay, thank you, Linda. Thank you for bringing the weather to come. I'm not allowed to move too far from the lactic because that's where the Zoom mic is. So shout at me if I want to go there. So I realized the other day that this title suggests there's going to be a lot of high concept computational stuff, and there really isn't. So don't panic, there's like 2 slides, but they're in the middle, you'll hardly notice them. Maybe the idea behind the modeling here is to skip some of the basic microbiology involved in drug discovery, and that's because basic microbiology, the learning of burgdorferi is really difficult. So we're trying to accelerate the drug discovery pipeline in an organism where doing genetics is difficult, doing metabolic stuff is difficult. Culturing is difficult, they just take forever to grow.
Apologies, we're having a little trouble hearing on Zoom. If you could get closer to the lectern.
Can't get that much closer. I can stand behind it, if that helps.
That helps significantly, thank you.
Okay. Oh no, that limits my wandering even more. Okay, yeah, so we're building this pipeline in Borrelia for studying Lyme disease, but what we're hoping is that we'll be able to use the pipeline to look at other difficult bacteria, so other things where genetics is difficult and metabolic stuff is difficult. Okay, really quickly, everyone probably knows this, but Lyme disease is largely treatable. We have antibiotics that work most of the time. Depending on how you count recovery, there's a certain percentage of people that don't recover after antibiotics. We don't entirely know why. It could be survival of the bacteria, it could be some kind of immune dysregulation. But we do know that in absence of good treatment options, people wind up getting treated with long courses of antibiotics. That's not really useful. There's no evidence that these long courses help, but some doctors like to prescribe them anyway. Again, long courses of antibiotics, as we all know, have all of the associated risks to the patient and also problems for society in general because of the evolution of resistance. So we think Lyme disease and Borrelia is a really good candidate for the development of narrow spectrum antibiotics, which would allow you to do these long courses without those associated risks. So is the point to work? No. Okay. Yeah, so if you have a narrow spectrum antibiotic, obviously you can kill the target organism but leave the rest of the microbiome intact, and you'll be kind of saving the broad spectrum antibiotics for where you really need them. Narrow spectrum antibiotics are still taking off. They're used for TB quite a lot because TB, you have to give these long treatments. That one at the top is used against C. diff and is relatively selective against C. diff. There's probably one or two more examples, but there's not loads and loads. I think we're missing a trick there because the use of narrow spectrum antibiotics would allow you to start thinking about infectious diseases in the context of precision medicine, personalized medicine, which is a huge growth industry, in contrast to antibiotics, which is not a huge growth industry. These are market projections for kind of how much money is predicted to be in these healthcare, business worlds by the year 2030. So antibiotics are stagnating, whereas personalized medicine is really going up. Note that antibiotics there is in billion with a B, and this personalized medicine is in trillion with a T. So I think if we can try and move ourselves away from antibiotics and sneak into this personalized medicine market, we might have more luck commercially. Okay, yeah, like I say, I think Borrelia we think is a really good candidate for the development of narrow spectrum antibiotics. Partly because of the clinical aspects, that we have to use these long treatments sometimes, and partly because of the life cycle and the genome of the bacteria, both of which are weird. So the life cycle, as you probably know, the bacteria shuttle around between mice and other small mammals. They're carried around by ticks, they get into larger mammals, occasionally they get into humans. What they don't do is ever live by themselves. There's no free living part of this life cycle, so they're always associated with a host, which means that, like a lot of host-associated bacteria, the genome shrinks. So the genome is tiny, 1.5 megabases. The genome is also really weird. It's got a chromosome, which is less than a megabase, and then a load of plasmids, most of which we have no idea what they're doing. These 9 are all a prophage, so they're not even doing anything. That's like 20% of the genome that's not really doing anything for the bacteria. So you wind up with a very small genome with very few coding sequences. And as a result, you have a very small metabolic capacity. So if you haven't seen these before, this is a metabolic map generated by KEGG. So this is an automated pipeline. I'm going to come back to how we can improve this later. But what we're looking at is dots represent metabolites and lines represent enzymatic reactions. So obviously Borrelia is just about alive, but it's really struggling. Whereas normal bacteria have all kinds of pathways. A lot of pathways do similar jobs, so there's a lot of redundancy. That's part of why a robust organism with a proper genome like Salmonella is able to survive in hosts and then in the wild, in water and whatever, is because they have a kind of flexibility. And so we think that because Borrelia has this really truncated metabolism, it's going to be particularly susceptible to metabolically active drugs. So if we can disrupt one of these pathways, then there's no spare pathway to compensate. I came up with this principle shortly after moving to Boston. Obviously, as we've probably discovered today, in a metro system with few lines, if one of the lines breaks, tough. There's nothing you can do. If you want to go from Tufts to Sam Adams at Stony Brook, then you take the orange line, but if the orange line is broken, then you just don't go. Where if you're in a modern, thriving metropolis like Paris, you want to go from the Eiffel Tower to Notre Dame, you can do the orange, or you can do the yellow, or you can do the pink and the green, or the whatever. So this is what I'm really interested in. It's basically all of the white space on the left, where there should be metabolism, but there isn't. Like I say, this pipeline sorry, this map was generated by an automated annotation system called KEG. It's the Kyoto Encyclopedia of Genes and Genomes. And it basically matches enzymatic functions to coding sequences from the genome and builds this map based on similarity to other enzymes. So the whole creation of this map is very dependent on the particular set of algorithms that are used to make the annotations. I wanted to make a better version of this so we can study it in more detail. So I basically started this process from scratch and re-annotated the genome. So when we're making, I'm going to try and move this. I can move that. So when we're making these maps, obviously you start from the genome. We're using the genome of the type strain, which is B31. In principle, you could do this with any strain. So we obviously could use it for other bacteria and do the same thing. Or we could also, something we're particularly excited about is going back to the personalized medicine thing. You could take whole genome sequence from a whole genome diagnostic, and run this through on a patient by patient basis. Oh no. I click on the wrong thing. There we go. There we go. Okay, so the first thing we have to do is annotate the genome. Borrelia is weird, like I say. It's down a little evolutionary arm, so annotating by similarity to known sequences is difficult, but it's the best way we have to do this annotation. So to try and cover as much ground as we can, I did 3 different annotation pipelines, of which KEG was one, but there's other similar ones. So Metapsych is the other big metabolic database, and the HMM there, the hidden Markov models, were a set of hidden Markov models trained on prokaryotic enzymatic sequences, and then we applied those to our genome and they made their own guess. You can do that with various kind of cutoffs and things. Again, what I did was basically run every combination I could think of and then pool everything together into one draft model. Having done that once, I then just did all of that again with the help of a summer student, Louise. So we have 2 existing pipelines, the new hidden Markov models in a bunch of variations, and we do all of that twice, and then we smash it all together into a draft metabolic model, which contains all of the predicted enzymes, all of their associated reactions, and all of the metabolites. The draft model has annotations from 6 different sources, and those sources are spread out over a series of months, so it's a real mess. So you go through this process, which is euphemistically called curation, which amounts to staring at the Excel spreadsheet for a long time and making sure everything joins up in the way it's supposed to. You have to unify the names, you have to unify the annotations. It's really boring and it takes ages, but there was a pandemic and the pubs were closed. So after the process of curation and cross-referencing to the literature, you'd wind up with a neat version, the final model. The final model I finished like 2 weeks ago, having started this in the pandemic. Although it's worth pointing out that the second time you do this, it's much faster. Making the first model is a long process, but then subsequent models, you can use the first one as a scaffold and it goes much faster. So we have this prediction. We cover 151 genes, which is not loads of the genome, it's 10%, but obviously we're only annotating core metabolic genes here, so most of it is not relevant. those genes encode 200 reactions. We need 85 transport reactions to make the system function, to input metabolites that are important down the line. And we have about 200 different compounds that are involved in the system. This is really to illustrate the value of combining those 3 different pipelines. So we annotated 208 reactions combined, but each annotation pipeline is kind of pulling its weight. So we get different ones from each pipeline, and then you go through that process of combining them all. So what we've got is a model here, which is more complete and more comprehensive than ones that have been generated automatically. It looks like that. It's an XML database file, or it looks like that, which is an Excel spreadsheet. Or if you really stare at it hard enough, you can make it look like this, which is an actual metabolic map, like the one I showed before, color coded here by kind of general pathway. So peptidoglycans over here, proteins are down here, nucleotides, etc. Yeah, so as a baseline, what we've got is a more complete description of the metabolism, the metabolic capacity of the organism than we've had before. And we can use that prediction of the metabolism to start trying to ask basic biological questions. So here, like Linda mentioned, we're interested in lipids because of another project. The yellow circles are all this is the lipid metabolism. And you can look at this and realize that there's no system for the bacteria to make fatty acids or make cholesterol. They just have to import cholesterol, which leads you to the prediction that those 2 should be essential nutrients. So we can make a growth medium without fatty acids or cholesterol and find that modified growth medium, they don't grow. And then if you add back in fatty acids and cholesterol, they do start growing again. So you sort of validate the basic metabolic findings from the model. Which is fine. You know, you can spend ages looking at these maps, and I have spent ages looking at these maps, but it doesn't get you that much closer to designing antibiotics. The critical thing for designing antibiotics is that as well as a pretty map, you've also got a mathematical framework that you can run simulations with. The maths is underlying all of this maths. There's some really complex stuff. I understand about half of it. And the half I understand is not interesting. So I'm going to skip most of the maths and give you a really quick kind of idiot's guide to what's going on, where the idiot in question, I guess, is me. So in order to run simulations, we essentially have to solve this equation. And the solution for these kind of things is you find out what the progress through each of these reactions is. So you identify how active each reaction in the system is under a given set of conditions. That's obviously a huge number of possibilities. So to narrow down those possibilities, we apply a set of constraints to the model to kind of narrow down the problem space and speed up the solution, the programming time that it takes to reach the solution. So some of those constraints are thermodynamic. You can't create matter, you can't destroy matter. Some of them are biological, so we know that some enzymes only work forwards and some enzymes are reversible. And crucially, we apply an objective function. So you have to tell the model what it's aiming for. So it solves the progress through each of these reactions in a way that maximizes one particular output. And in our case, that output is growth, is the generation of biomass. There's also something called a matrix. I stopped studying maths when I was 15, so I have no idea what a matrix is, but it solves in MATLAB, where mat, it turns out in MATLAB, means matrix. MATLAB does its thing. The thing is called flux balance analysis. It balances the optimum flux through each reaction. And you wind up with a simulation of growth. Obviously, it's in heavy inverted commas because it's in silico. What we're really testing for is the production of biomass precursors, which is this, the inner ring here of all the things that are components of biomass that have to be made. So we can make this system grow in inverted commas. Obviously, we're not that bothered about the bacteria growing. What we're really interested in is them dying. So having spent like a year getting to this point where we've got the system growing, we immediately start breaking it again and ask which of these reactions are essential to that growth process. So which ones, if you eliminate that particular reaction, which ones prevent growth completely? Again, maths is involved. And then you get this prediction where all of the essential reactions are in pink, and the essential reactions are obviously drug targets because they're things that kill the growth phenotype. So the metabolites in pink are all the ones that we need to make biomass, and the reactions in pink are all the things that are essential to make those biomass precursors. This is a more user-friendly way of looking at that. So I'm almost at the biology. These essential reactions are drug targets. There's 77 of them that are predicted, which is a relatively high percentage of the whole model, but then it's a small model, so you expect more of it to be essential. And obviously it is a model of core metabolism. So again, it's sort of there's none of the kind of accessory stuff in the model in the first place. 77 reactions is its genes, some transport reactions. We have a transposon library already. The library isn't saturated, so you can't use it to predict essentiality. But we do know that anything where there is a transposon is probably not essential because it can be disrupted. And we match that prediction reasonably well. I actually thought that was okay, 90%, but then you look in the literature and some people seem really happy with 60%. So I think as the field goes, matching the transposon library by 90% is not bad. I'm not going to give you the boring details, but we can look at this and then look at what's been studied before, and we find that enzymes where they've had to make conditional mutants because they're synthetic lethal, we recapitulate some of those phenotypes, and we find in this list of 77 metabolic reactions, we find some things that have been described as drug targets before. But I mentioned at the start that I think what we're really interested in is narrow spectrum drugs. So in order to find those, what we've done is taken existing models of E. coli and Staph aureus. Someone else made those, so I didn't have to do them. And crucially, I also don't have to talk to you about them. Someone else has made those. These are much bigger, but you can do similar analyses with them. So you can find the essential reactions in E. coli and Staph aureus by the same pipeline. So if we cross-reference Borrelia with E. coli and Staph aureus, which we just chose because they're kind of an easy model gram-negative and an easy model gram-positive, we throw out 32 unique drug targets, so things that are only lethal in Borrelia. This process gets rid of a lot of the things that are, drug targets, but not interesting ones. So things like the cell wall, which we've studied to death, and translation, which we already know that those are drug targets. So we've got 32 unique enzymes that we can start to try and inhibit. What we've started with is existing inhibitors, so things that are available commercially or existing drugs. And the aim really is not so much to put those into clinical practice immediately, although that would be great, but the aim is to use the existing compounds. If we can get any kind of activity at all, we can use them as lead compounds to try and improve and develop. Obviously, most existing drugs are designed with human enzymes in mind, so they're going to need some work to get them to target the Borrelial enzymes effectively. So the first one of these we tried, that's a lie. One of the ones we tried that worked was pemetrexed, which is an anti-cancer drug. And it inhibits this reaction. It makes methylene THF, which is an important cofactor in a bunch of things, including ribosome maturation and translation. And we find that it kills Borrelia fine. The MIC is about 0.5 milligram per mil. The unit has been skipped off there. I could have told you anything. I could have told you it was 0.5 microgram and we were basically finished. It's 0.5 milligram per mil. Obviously, the important thing that we're looking for is activity against Borrelia, but not against E. coli and Staph aureus here. And so this drug does have a degree of selectivity. We don't know, obviously. I assume if you push this out super high, then you would start killing the others, but certainly in this concentration range, we get a selectivity. We have several examples of this. I'm not going to show you them all because the graphs all look the same. But theophylline is actually, theophylline was used against asthma, and it was used for kind of long treatment. So it's probably more tolerated by humans than the anti-cancer drug. Pyridoxal phosphate is a cofactor in all kinds of stuff. A bunch of enzymes use pyridoxal. It's vitamin B6. And theophylline inhibits the production of that phosphorylated version. And again, we see selective activity. We're still working out this exact assay, but in addition to inhibition of growth, we can get these drugs to give some degree of killing of stationary phase cells as well. So this is a microscopic assay. We basically have a green stain which stains all cells and an orange stain which stains just dead cells. So you can count them up and you count the green ones and you count the orange ones and you know how many of the cells are dead. We're working on the details of this, and by we I mean Keely. But so we're killing sort of some of the bacteria, which is fine, but then doxycycline, the current clinical drug of choice, actually doesn't kill any of them, or doesn't kill any more than there's always a certain amount of the healthy population that are dead. So doxy is kind of bacteriostatic, but still seems to work. So these we've got some killing, which is better than doxycycline. The reason we think these drugs are selective, we can go back and cross-reference the various metabolic maps. Borrelia imports pyridoxal and then phosphorylates it. You can probably guess where I'm going. Staph aureus can do that, but also has its other path. So that inhibition of this enzyme here is not lethal to Staph aureus. E. coli has a similar pathway there, but it just different specifics. It involves threonine somehow. I think the thing that was really gratifying was this. The top two graphs here are pemetrexed and theophylline, the ones I showed already. The bottom two are two other drugs that were predicted to be lethal to Borrelia and also variably lethal to E. coli and Staph aureus. So donopyruvate is a glycolysis inhibitor. Borrelia is dependent on glycolysis and doesn't have an electron transport chain. Staph aureus is also dependent on glycolysis. E. coli is not, and we see that the drug kills Borrelia and Staph aureus, but it doesn't kill E. coli at these concentrations. Cycloserine at the bottom. Cycloserine at the bottom inhibits alanine racemase. D-alanine is obviously a precursor to cell wall biosynthesis. So all bacteria need to synthesize a cell wall. So we see that cycloserine is active against all three compounds, against all three species. So just as a slightly easier way of looking at them, all those graphs. This is just side by side. So the predicted growth rate here, if the growth is 0, then essentially then the model is predicting an absence of growth. And we see that where the model predicts lethality, then we do reach an MIC, and where the model predicts that there's no effect, then we don't reach an MIC. We've tested a bunch of these. I've tested 16 compounds, I think. Some of them work and some of them don't. What we've got in terms of anti-borrelial activity, we're at about 40% of the compounds we test work, which is giving us plenty of leads. It's also obviously way better than doing a compound screen where you're screening 50,000 compounds and maybe you get 1 or 2 hits. So it's not perfect, but I think it is finding as lead compounds, and it is enriching and it's speeding up the process of identifying those lead compounds. So this is kind of where we're at. Obviously, there's a bunch of work still to do. Identifying the leads has always been the easy bit. Developing them is a longer process. What is it? So what we're trying to do is obviously nail down the mechanism. We've got the predicted mechanism, but we haven't validated it here. All we've shown is that the compounds are effective. So the first thing we want to do is to nail down whether the predicted mechanism is the actual mechanism of toxicity. And we're going to do that in kind of traditional microbiological ways. So we can sublethal challenge and then see what the transcriptional response is, and that gives you a clue. We can also try and raise resistant mutants and sequence the resistant mutants, and that should again tell us. It gives you a clue. I guess it doesn't tell you the exact mechanism. We're also going to look in vitro, and I think this is the gold standard proof, I think, for mechanism, is we'll purify the enzyme and then see if the compounds can inhibit the enzyme in vitro. This will also be the beginning of the process of optimizing those lead compounds. So once you've got pure enzyme, you can get a crystal structure and you can try and design improved versions of the compounds we've found. There's also obviously the animal work. Animal work is complex in Lyme disease because there's a lot of animals involved. So you have the mice and the ticks as the two kind of model organisms, and then there's a lot of different steps at which you can try and intervene. So you can try and you can treat the infection in the mice, you can treat the infection in the ticks. You can stop the infection moving from the one to the other. So there's a lot of combinations of the animal work, and we need to decide which ones of those are the most likely to be useful. I'm really interested in prevention. I think if you could find a drug that's well tolerated, and you can give it to high risk people, it's a lower barrier. And obviously, if you're trying to cure an established infection, then you've got to distribute the drug all through the body, and you've got to get high MICs all through the body. And where if you're trying to get prevention of the transmission, then you can you're trying to kill fewer bacteria, and they're all in one place. I think that's the thing I'm really interested in, is prevention. So, like I said at the start, I think that we've got the possibility of trying to use this pipeline for other bacteria as well. And obviously, you know, with a lot of bacteria, you've already got the genetic tools and you can just do this by microbiology, but that's not true for all bacteria. Borrelia is host associated, it's got a small genome. We think that makes it vulnerable to this kind of metabolic disruption. And it's also difficult to work with in the lab. Those four things are obviously not a coincidence. They're all related, right? They're difficult to work with because they're fastidious, because they have a small metabolism, etc. So I think this is kind of like a subcategory of pathogens, is these ones that are particularly fastidious and hard to work with. All bacteria in the world are mostly unculturable. It's mostly because we haven't tried, because no one's that interested. But even pathogens, there are about a quarter that we can't culture in the lab. And even among the remaining 75%, there are plenty that we can work with, but it's a pain in the ass, like Borrelia. So those are the ones we think this kind of pipeline where you're skipping the really basic microbiology might be useful. This is a small list of those. I'm interested to see if anyone else has any ideas. I think we have, these aren't all bacterial, but so we have things that are impossible to culture, and we have things that are difficult. There's loads. Obviously, this way of doing this way of predicting essential reactions could also be useful just for the species where we need more drug targets, right? Or the high priority pathogens. It's just a different way of identifying drug targets. So, you know, things like Neisseria and MRSA and all the escape pathogens and stuff. I think just as a discovery tool, maybe it's still useful there. So this is what I'm imagining as a pipeline. And part of this is a research pipeline, and part of it is also an implementation pipeline. Like, you could actually do this in real life, I think. We can go from a genome sequence to mapping the essential reactions. The genome sequence, like I say, ours was from a model organism, but you could take whatever genome sequence you like. And once you've done the mapping, then you've got the process of small molecule screening and refining the leads and producing the drugs. But obviously, once you've done this a few times, you can skip that step and you can do the modeling, make the predictions, and then, you know, go, oh, well, we've already got a pyridoxal kinase inhibitor, so we can just skip to the good bit, and you know what drugs might work. Just as a last thing, obviously the biggest barrier to making new antibiotics is not really scientific, it's financial. The way all these personalized medicine companies work, and personalized medicine is a huge industry in cancer particularly, you make money because you're selling a pipeline rather than a drug. So you're selling the service of doing the sequencing, making the right diagnosis, choosing the drug, and then giving the drug. So you're essentially de-risking each of those aspects of your company by having them underwritten by the others. And I think that's, again, it would help. It would be an alternative to the current way of designing antibiotics, which is where you sink billions of dollars into a lead compound, and then if it works, you just go out of business because you can't make any money off it. So here, these cancer personalized medicine companies, diagnosis is its own business. You also generate loads of data, and we can discuss the ethics of selling that data, but they do. So they make about half of their money on the sequencing and the data from the sequencing. So the fact that you're making IP and the fact you're selling drugs to people is kind of a nice bonus. You've already got a functional business doing the first bit. That's obviously a long way off, and we need to make some compounds first, but I think it's a really, I think it's an interesting idea of how you might get around the big bottleneck in drug development at the moment. Right. Which is where I'm going to leave it. Thanks to Lyndon and everyone in the WHO lab, especially the people involved who actually did some of this work. The three tools there, Cobra, Raven, and Cytoscape, are what I use to actually do the modeling. I am not clever enough to have written that code myself, so that's all come from them, funded by the NIH and a few other people. Thank you. Thoughts, feelings?
The validation for it, did you also, since you know the metabolic disorders,
yes, so we could overexpress the enzymes to validate. One of the things I really like about that pathway is that you're not doing any cloning in the target organism, which I think is important for two reasons. Obviously, well, the first one, I don't like cloning, but the second one is that we have some cloning tools for Borrelia, but we don't have cloning tools for other bacteria. So I'm trying to do this in a way that you could port the pipeline over to rickettsia without having to do the genetics there. But yeah, I think you could. One thing I'm interested in is the sort of quick and dirty version of that, where you can spike in the product of the enzyme, right? So in theory, say theophylline, the pyridoxal kinase inhibitor, if you spiked in loads of pyridoxal phosphate and could recover growth, then that asks the same question, but in a way that's much easier. So well, yeah, I don't know. It's a really nice experiment if it works. If it doesn't work, it's inconclusive. So yeah, I think so. That's definitely something we could. And I think for the first pass, in Borrelia, maybe to really make sure of the mechanism, we could do that. But I think as part of the pipeline, I'd like to not do cloning in the targets. Oh, wait, is there some in the chat? No.
Yes, we have some questions in the chat.
Can you take a bug in a drug and retrospectively test? So figure out whether fidaxomicin is specific because of a metabolic thing. I think you can. I think in fidaxomicin's case, it's actually, I forget the target. I think it's something ribosomal, but I think it works. RNA polymerase.
Like the cell structure.
So it might be RNA polymerase, but yeah, I think the thing is that the compound there only binds to the C. diff version of the target enzyme. So I think you could, but the answer isn't always going to be that it's a metabolic weakness, right?
Yeah, I guess the question is whether or not there's some other pair where you might not explain the selectivity on the basis of drug recognition, but on the actual pathway that it interrupts.
Yeah, I think you certainly could, and you could certainly, you do the essentiality prediction and then spend an afternoon just thinking about it really hard. Yeah, I think if the answer is metabolic, then yeah, I think you could find it in that way.
Thanks.
What's the basis of the green versus the yellow in the killing assay? So the assay is called Backlight. It's a commercially available one. The green dye is membrane permeable, so it enters all cells. The red dye is membrane impermeable, so it only enters cells that are dying and starting to fall apart. So it's indirect, and it's really an assay that measures membrane integrity rather than death, but death causes a loss of membrane integrity, so it's kind of a proxy. I don't know, Ian.
Well, I guess you gave it a shot. Can you hear me?
Yes.
Oh, perfect. Thanks so much for this talk. My name's Ian DeStefano. I'm a veterinarian in the vet school. I just wanted to make a comment that this is super cool, and just throwing out there that, if some of these drugs are in fact validated through the whole pipeline process in the in vitro model, et cetera, that you outlined there, I'd urge you or some folks from your team to reach out to us, because dogs get a lot of Lyme. We see a ton of Lyme in New England, and we could easily use them as a translational model. I'm particularly intrigued by the potential idea of theophylline, because that drug is super easily tolerated in dogs, and we use it. It's pretty crummy bronchodilator in dogs, but we do use it on occasion. That would be super cool. So keep us in mind.
Yeah, absolutely. I will. Thank you. Yeah, again, the dogs and other animals are also important commercially. People spend a lot of money on their dogs, so I think getting a drug approved for vet purposes would also, financially it would be good, and it would help you keep going into humans.
This is John Leong again. I just will mention that Brian Noonan and I met with Karina Beal at the vet school, who also proposed being able to use the companion animals, for example, as ways of furthering drug development. The regulatory bar, of course, is much lower than it is for human drugs.
Yes. I also want to test the centric diagnostic test that we're developing on dogs. So I'll try and speak to some people. Have the metabolic analyses thrown up any other interesting stuff, surface proteins? So no is the short answer. The metabolic map really predicts only kind of core metabolism. So things like surface proteins are not predicted. So we basically don't touch the surface proteins. That is I guess a weakness of the model, in that you're only working with core metabolism, right? There's probably loads of drug targets that aren't core metabolic that we miss. It's partly because it's hard to do mathematical predictions with surface proteins, like how they contribute to viability. Partly just for the sake of keeping these things manageable and not needing a supercomputer to cover everything. The model is restricted to core enzymes, basically. Question from one of the... Is DNA analysis based on... Oh, okay, yeah. So currently we're using the type strain B31, which was isolated from a tick 25 years ago. But yes, in principle, I think we could do human isolates, or rodent isolates, or tick isolates, or whatever. And like I said, once you've established the pipeline, it should be faster the second time around because you can use the first one as a scaffold, which would give you the option of doing this on a personalized basis and looking for particular strain to strain variations in like particularly difficult cases or whatever.
Well, the problem is that the lab-grown strains chuck out plasmids, linear and circular, and you might be missing several pathways that are present in the actively infecting bugs.
Yeah, yes, that is a problem. From my analysis of the type strain, there's not that many core metabolic genes that are encoded on the plasmids. So if you're lucky, they might throw out plasmids that aren't relevant to the model. There are a few, obviously. So yes, those could be missed as you passage and lose plasmids. If you were able to do direct sequencing in a way that's not viable yet for Borrelia, but I think that's the direction of travel for infectious disease diagnosis, is just having everything whole genome sequenced and then picking the noise, picking the information out of the noise. So I think if you're able to do it on direct whole genome sequence, you get around that problem of losing the plasmids as you passage. All right, thank you.
Thank you so much, Pete.